thank you all for tuning in and coming to the class. So my name is Matt Frieman. I am a virologist here at University of Maryland. And my lab works on coronaviruses. We've done this for years since I've been here. I've been here for 10 years. And then I worked on them for about 5 or 6 years before that. So I started working on the SARS coronavirus right around 2003 when I started my postdoc at North Carolina. And so what I wanted to do for the talk today was to give you a bit of an introduction to this virus that we're in the middle of now, and really go through. I made a list of kind of 10 kind of major questions that I thought everybody might have to kind of tell you about the virus, what it's going on right now, and then where I think it's going in the future. As you said, everyone can type in questions in the chat box if you'd like. If there's some that I see that pop up during the talk that make sense of getting them as we go, I'll jump on them as I go through the slides. But if not, I'm happy to answer as many as you have at the end of the talk as well. All right. So for me, the goal of the talk is to really provide facts as best I can about the current outbreak, to give you the kind of the language and the understanding of the virus that causes it, this new coronavirus, and then really explain what my lab is doing for this outbreak, just a little bit about our drug work, and we're in the midst of a lot of things at the moment. And then really at the end discuss where we're going from here and where I think the outbreak is moving in the future, and what we should be discussing and what we should be worried about, both as as citizens in this outbreak, but as also kind of parts of it as we are in Baltimore. All right. So just to start, the quick kind of nomenclature guide as we'll go through the slides. So as you all know, this virus, we'll go over this in a second, but the virus emerged in Wuhan, China. Initially it was called the Wuhan coronavirus, and then the press calls it the coronavirus, even though I'm going to teach you that there's a lot of other coronaviruses out there that infect people. The WHO officially calls it COVID-19, which stands for coronavirus disease from 2019. That's what I think the general press has been calling it now. So we hear about COVID and COVID-19. And then there's officially the name is SARS coronavirus 2 or SARS-CoV-2 from the Virology Institute that names official viruses. And so we'll talk about SARS-CoV-2, COVID-19, the original SARS virus, a little bit about MERS, and then the other human coronaviruses that we know of that give us colds every year. Okay, so first, what is this thing we call COVID-19? So there are multiple human coronaviruses that we know of. There's 7 now. There's 4 coronaviruses that give us a common cold every year. So we get infected with coronaviruses every winter that give us just a runny nose and a mild cough, and that's it. The names of them are shown. You can see my pointer. The names are shown here. They're not that important, but just to know that we actually do get coronaviruses every year. They give us a cold, they resolve, and they go away. There's very little research done on these viruses because they're kind of mild cold viruses, and it's really not something that people study that much. But hopefully in the future, now that we know about this new virus, they will be. The other viruses that we know of in the past that really are more concerning and highly pathogenic are SARS coronavirus and MERS coronavirus. And so SARS stands for Severe Acute Respiratory Syndrome. That emerged in 2003 in the Guangdong province in China. MERS is the Middle East Respiratory Syndrome coronavirus. It emerged in Saudi Arabia, or at least we first identified it in Saudi Arabia in 2012. Both of them are highly lethal viruses. SARS has about a 10% fatality rate. It only infected around 8,000 people around the world. It's no longer SARS-CoV-1, the one we know about from 2003, is no longer in people. It spread and then died out in about 9 months. MERS coronavirus has affected about 3,000 people, but it kills about 35% of those infected. So has a much higher fatality rate than SARS or SARS-2. It's a quite lethal virus, but it only infected so far around 3,000 people. So it's not as transmissible. And then this new one, SARS coronavirus 2 or COVID-19, is kind of in the middle of the seasonal coronaviruses and their lethality and the regular coronaviruses, the high path coronaviruses, where the lethality is somewhere around 1 to 3%-ish, depending on the population. All the coronaviruses look like this on the picture here. They kind of look like a big soap bubble with a lipid envelope on the outside and then these spikes that come out from the outside of the virus. This is what the virus looks like. When a cartoon on the right is what a electron microscope picture of it does. You can see these spikes that come out of the virus, and those spikes are what bind to cells on your lungs, and then give you it gives the virus the ability to infect the cells. It's also what all the vaccines are directed to. We'll talk about that at the end. But all the vaccines that are being developed now are directed against the spike protein on the outside of the virus, because that's where the antibodies need to go so they can block infection. I know this is more sciencey, and I'm trying to give it a more general talk here, but I just wanted to show you, you don't have to know all the names of these viruses on here, but this is kind of the spectrum of coronaviruses that we know of in people. This is from several years ago. You can see that the only thing I wanted to show you here was that where the sneezing girl is, these are the 4, and the red arrows, these are where the 4 human coronaviruses are that just give you a cold, with SARS and MERS being in this blue group up here. And then this new virus is quite similar. So this is a tree that we make looking at comparing the genomes of all these viruses. And the point here was just to show you that the new coronavirus, SARS coronavirus 2 or COVID-19, is quite similar to the SARS coronavirus from 2003. So, and so it's important to understand kind of the dynamics of these things, and the regular coronaviruses that we have right now are not related directly really to the coronavirus that's spreading. It's also important to point out that the regular coronaviruses that we get every year cause around 10 to 40% of our colds during the winter. So everybody here who's listening has had one of these coronaviruses, or all of the seasonal ones, these 4, the red arrows, before, and we all have antibodies to them. And we don't really know why we get colds every year from these viruses, even if we have antibodies. So there's a lot of questions that actually go along with the coronavirus understanding for years. Okay, so that's what this virus is. The next thing we'll start with is, where did this new virus come from? And so if we go, it really started that we know of in the world. The first announcement of it was on December 30th and 31st last year. So really not very long ago. There's a website called ProMed, which lists all of the communications around the world for new and emerging viruses and diseases. And so there was this announcement on ProMed in December 30th and 31st that there was an undiagnosed pneumonia, a cluster that was occurring in the Hubei province in Wuhan in China. And so this was associated with a seafood market initially, which was quite interesting because the original SARS emerged from bats through something called a civet cat in these animal markets in China. MERS, the other coronavirus from primarily in the Middle East, is in camels, endemically in camels, and spreads from camels to people. And so we're always looking for these animal to human jumps that happen all the time for all kinds of viruses, not just coronas. But this was kind of an interesting thing that was maybe something linked to an interesting virus. At the time when this was announced, there was just a cluster of pneumonia cases, upwards of 41 cases by the January 1st of 2020. And so on the 31st, it was announced there was 27 people linked to this Huan seafood market in Wuhan, and the WHO was alerted, and then all of the countries around the world were alerted to look out for some types of unknown, undiagnosed pneumonia around the world. Right away, the market was closed and disinfected and shut down, so nobody could get contaminated potentially from that market in the future. It was about a week later that the initial virus was identified as a coronavirus. So they took the virus out of these people who had pneumonia, they sequenced the virus, and then they compared its genome to all the other known viruses, and it was clear that it was a coronavirus. And then a couple days later, the sequence was put online, and we had clearly identified that it looked like SARS from 2003 in the same kind of cluster. It was in that same kind of red arrow that I showed you before, where it looks like another coronavirus in the SARS grouping. And what's been known since 2003 was that when people started looking for coronaviruses in bats, especially in China, they found a lot of viruses that looked like SARS, but were a little bit different. So they were all related in some way, and then they found a lot of these viruses in bats. And bats are a harbinger of viruses all around the world. They seem to live with them very simply, and they don't get disease from most of these viruses, other than really rabies. And And so SARS has been shown here, MERS as well, Ebola, Marburg, Nipah, and Hendra are two other viruses in Australia and Southeast Asia. And so bats have a kind of a remarkable biology, which we won't go into. But initially, it was identified that this virus was a bat SARS-like virus. And then the same kind of electron micrographs that I showed you before were done for this new virus. This is a picture from John Birnbaum at NIH. And it looks like the same coronavirus that I just showed you before. Okay, so that's where it came from. The next idea was, we knew it came from China, but what did it really come from, an animal or another human, or what do we know? And so, as I said, most of these coronaviruses originated in animals. All the ones that we know of in humans originated in animals at some point. We don't know what the animal reservoir is for this new virus. It probably spilled over, as I said, from bats into an intermediate animal and then jumped to humans in the fall. And so what is very clear is that all of the viruses that came out in these initial people that had pneumonia were all identical viruses. And so what we know from previous outbreaks is that as viruses jump from animals to people, they have to mutate a bit because the animal version of it doesn't really replicate that well in people. But what was found is that if we look on this tree here, which you don't have to know anything about this other than look at these red ones down here, they're all 100% identical to each other. And what we clearly know is that all of these viruses all of the ones that were in the people initially in this outbreak were identical. And they replicated incredibly well. They didn't have to adapt to humans. They didn't have to have any mutations. They worked well and they started spreading very rapidly. And so this virus was probably very human adapted, and it had been around for a little bit longer than just the end of December. Okay. Again, this is just a cool picture I wanted to show you. One of the kind of amazing science things, and I think you heard from Jacques Ravel last week about the sequencing we can do for all kinds of pathogens. One of the amazing things that's going on right now in the context of this outbreak is that the viruses are being isolated from people and then being sequenced incredibly rapidly all around the world. And so this is just a picture, a way to kind of analyze all of these virus sequences that are coming out. There's an interesting website called Nextstrain, run by this guy named Trevor Bedford at the University of Washington. And so each one of these lines and each one of these little dots is a virus that's been sequenced from a person. And so what we can just broadly identify from this is that there are viruses that started in China and then spread to the rest of the world, both Europe and then United States. If you look just in the United States, if you look at these red dots here, all of these viruses now cluster together that are coming out of Washington State. So 83% of these viruses, and we can track back the sequence, originated from a single person that was infected in Washington State and then spread the virus to other people, really unbeknownst to anyone else as it was spreading. One of the really other kind of amazing things we can do with the sequencing analysis of this is that by comparing all of the little tiny changes that occur in the genomes of these viruses, you can follow them around the world. And so this is data looking at the United Kingdom. What you can find is that there's been a bunch of different countries or people from different countries that have brought their own strain essentially of the virus, their own isolate, into the UK. And so unlike the Washington State case in the United States here, this looks like it originated in China. If you actually did this for New York, the New York cases probably originated from Europe. That's what the sequence analysis tells us. But in the UK, we have multiple different isolates coming in from all around the world in different people as they originate there. And I think the important point I want you to take away from those kind of rainbow pictures is that while in the news we talk about different strains of the virus moving and there may be being different strains and sequences around the world, they're all essentially the same. And we are all being infected by the same virus. And that there's no evidence that any of these differences in the genomes are making it more lethal or less lethal right now. And so this may change in the future, but right now, all of the viruses are essentially identical to each other, and nothing is kind of winning and making it worse or better as we go. Okay. So question 4 is, what are the symptoms of COVID-19? So I think most of you have heard about all of these in the news, but they basically look like an influenza-like illness. They look like a cold. And so they give you, the general symptoms are cough, fever, feeling tired and malaise, shortness of breath, difficulty breathing, sore throat, and general body aches. So normal kind of respiratory disease that you would know of before. In some people, there is this clear link to GI symptoms, so they're having diarrhea or other stomach issues, as well as this really interesting loss of smell, so called anosmia, where what we think is going on is that the virus is infecting the cells in your nose, and your nerves that give you smell from the nerve to your brain, they connect to basically the back of these cells in your nose. And so if the virus infects those cells and kills those cells or makes the kind of inflammation and immune response, then you can lose smell and lose that signaling. It seems to come back in several weeks, but there's an interesting link to this loss of smell issue. In recent reports, there's a lot of news about blood clotting issues and people getting blood clots and strokes based on during their infection. And I don't know what the cause of that is yet. It doesn't look like we really understand exactly what that means. The other really important thing here is there's a lot of comorbidities that are linked to more severe disease. And we'll talk about more of this in a second, but basically being greater than 60 years of age, having other comorbidities like diabetes, heart disease, any other type of thing that makes you immunocompromised, all linked to severe disease and more death if you get infected with this virus. So it's very important for anybody who has, from the elderly population, from that has any other immunosuppressive conditions, to really protect yourself from getting this virus and until we know more about it and have other therapies that we can treat people with. If we look in the United States, this is a graph from the middle of March, but the data is the same now, showing the number of hospitalizations in light pink by age, as well as ICU and case fatality rates. And I think the important thing here is that it's not just 65 and older that have severe disease. There's as many cases under 50 as there are over 50 in the hospitals. And so young people are not immune to this virus. They still get infected, they still get severe disease. They still have comorbidities like diabetes and obesity. And it's not, just because you're young and you're 20 and 30-year-old kids, you don't get a pass for this virus. The other interesting thing here that's been shown really around the world is that kids around 10 and under don't have severe disease at all. They tend to be very asymptomatic. This is what led to a lot of the school closings, is because kids can get infected. I have a 10 and a 7-year-old at home, or I guess 11 and 8-year-old now. One just had a birthday, and they can get infected. And even though they don't show symptoms, they can then spread that virus to their friends and family and grandparents and aunts and uncles. And so they may not have severe disease, they might not have a runny nose or cough or anything like that, but they still can spread the virus. And we do spread the virus by just breathing and coughing. And so it's really important that we control all of those droplets. Okay. All right. So next question is, how many real cases are there of this virus in the world? So there's a bunch of websites that are up there. Hopkins is a great website. The New York Times is a great daily website of this with continuous reads on this. But this is just a picture from the Hopkins website that's been really cataloging these cases from the beginning. Worldwide right now, we're over 3 million cases, which I just find to be completely remarkable. There's a little over 200,000 deaths worldwide, with a worldwide case fatality rate, if you just divide the deaths by cases, of around 6.5 to 7%. It's a very high case fatality rate. It changes in populations and countries, but it's an important aspect of this, that it's not just the flu. We are getting many, many more deaths around the world from this virus than we do from flu, and the rate of these deaths is much faster. This is a little video I grabbed from this other website called Our World in Data. It shows kind of the rate of growth of infections from these countries that I selected around the world. In red, you can see the United States, and this is the way this graph works. Is it's starting the counting from the days since the first 100th case in the country. What you can see is that United States has the global leader in number of cases around the world. If I go here, I'll go back a second and rerun this thing. There we go. China clearly had the first burst of cases. Once they initiated their mass quarantine measures that really flatlined their caseload. They still have cases every day now, actually, from imported cases, but it's not as clearly not as big as it was before. In Spain and Italy, you can see they had another massive spread, basically doubling of cases every 2 to 3 days, but now leveling off. And the United States cases, while still increasing every day, are starting to show a level off here. We haven't started a downturn on the number of cases. I'll show you in a second. But there are other countries that have done a really, really positive job at this. So South Korea and Singapore are the two that are the lines that aren't labeled here. They have had a much steadier and slower spread of this virus because they really started quarantine measures and case tracing right at the beginning of the outbreak as we started knowing about it. They also have a lot of infrastructure in their countries because of SARS that occurred in 2003. They've started much faster, much rapid, widespread testing than we have here. If we look in the United States, this is a map from yesterday showing where all the cases are around the US. So there's over 1 million cases so far with around 56,000 deaths, around a 5% case fatality rate. So we're matching basically what we're seeing around the world. Just yesterday in the United States, there was 24,000 new cases and 1,301 new deaths. So this virus is not going away. And I will make a plea later, but my plea now is that we are, all the social distancing measures and the quarantine measures are, we're not ready to dissolve those yet around this country at all. I know they're starting to dissolve, they've started them, and especially in southern states in Georgia and South Carolina and Florida, to release some of these quarantine measures, but it's definitely too early. We're still on the upswing of this virus. If we look at where the hotspots are, this is taken from a couple weeks ago. I grabbed this, I made this video from website data as well. What you can see is that as the initial cases started in Seattle and New York, there's been additional hotspots around the country in Boston, Detroit, Chicago, especially Baltimore, DC area, Southern Florida, New Orleans, and Louisiana, California. And so there are cases that are still accruing around the country in all of these areas. And hitting the healthcare centers really making a much difficult process to handle these cases in the hospitals. If you look at the number of reported cases that are day-on-day changes, so this is the number this is where we are now as of the 26th, the data I could get last night. You could see this through March, there was this really exponential curve in the mass number of cases that were accruing in the United States on a day-to-day basis. This was really both at the spread of the virus was important here, but also the testing and the PCR testing that was being done in the hospitals was ramping up here. So both of these kind of data sets and differences are baked into these numbers. Right now, there is still not widespread testing around the country. You can't just go get a test if you want to test. There's many, many people that are at home that have mild symptoms that didn't go to the hospital, which is the only place you generally can get a test. So the numbers are what the numbers are. There's clearly 5 to maybe 10-fold more cases around the country than we know of at the moment. But what this does show, at least at the current testing levels, is that the number of new cases are not diminishing. We're basically flatlining right now around 30,000 or so new cases a day. If we zoom into Maryland, so far, as of yesterday, there's been a little under 20,000 cases in Maryland with 945 deaths, really scattered all around the state, including the Baltimore metro area and around DC, where the hotspots are. Really, the county or the district with the highest number of cases is right near us in Baltimore. Right in the Park Heights, Ricetown Road area is where the largest number of cases in the state are. And it's unknown exactly what the cause of that burst in cases there is. If we look in Maryland itself, looking at number of new cases here, we are not on the downturn. So we'll go over what the measures would be to start opening up the state again that have been posed by Governor Hogan. But in Maryland right now, our caseload is not going down. If anything, it's trending up a little bit. This red line is the average of the last 7 days, and the pink lines here are every day's numbers. And so while yesterday may have had a little dip in the number of total cases by a couple hundred, going from around 1,100 or 1,200 or 100 on the 25th to around 800 on the 26th, we are not seeing a really steady decline yet. And this is it's a big worry across the state, and we still need to continue these social distancing and quarantine measures to protect our population. Okay, So that was our numbers, which aren't the most positive way of doing this. Let's talk about tests. So this is another really important aspect of this outbreak, is that the testing measures that were accrued in the United States, starting with really a kind of a problematic test that rolled out from the CDC when they scaled it up, has really delayed the testing and has caused a big problem. So I won't go into the testing problems. We can talk about it at the end if you'd like. But these are the two types of tests that are being used in the United States. And so the first one looks at the viral genome. So when we hear about all the testing that goes in the hospitals, it's really looking at the genome of the virus. So they swab your nose, they do a nasopharyngeal swab where they kind of reach way back in your nasal turbinate. So these the cells and the folds that are in your nose. So they use a really long probe, not really long, it's a deep probe into your nasal cavity to swab where the virus is. And then they take that swab, they put it into a reaction in the lab, and they test to see if you have replicating virus. So is the virus in your nose or not? And it's a yes/no process. The Institute of Genome Sciences here at University of Maryland, actually, Jacques Ravel, who you heard from last week, he's running a new testing center here for the entire state, and they want to scale up to 20,000 tests per day run for the entire state here at University of Maryland. So this is really, it's called a PCR test. It amplifies the genome of the virus, and that's what you get, a positive or negative on these responses. New tests actually show that saliva may work as well, and maybe there'll be a future rapid test for saliva. The turnaround for these tests is generally about 24 to 48 hours, depending on where you are in the state and in the country. The new tests that are coming online are looking at antibodies. And so just a little brief idea of what these serology tests are. So this is called a serology test. It's looking to see if you've been infected in the past. So just like we have antibodies to all the viruses, whether it's flu or mumps or measles or whatever other virus that we've been infected with in the past, we have antibodies to them in our body. And so if you have, you can use a serology test to determine if you've been infected with this virus in the past. And this is basically a blood sample. You can use a finger prick, usually for most of these samples. It looks at the serum, so where the antibodies are, and it will tell you whether you've been infected or not. And that's ideally what these work on. There's a lot of concern about these tests. These are not FDA approved. There's a couple hundred or so different companies that sell them in the United States. They're not regulated at all. And I would caution anybody who thinks that he buys these tests and does them at home, because we don't really know what they report. There's a lot of questions about false positives and false negatives. So do the tests tell you you really have antibodies when you don't, or does it not tell you don't have antibodies when you really do? But more importantly, having one of these tests be positive does not mean you're protected from being infected again. We don't know what these tests mean. We don't know how much antibody you need to be protected from being reinfected with this virus. We're at the early days of this right now, and I caution everyone, if they're looking or they're offered serology tests, to be very wary of what they mean. We don't have any idea really what they mean yet for this virus. Okay, so question 7 is how do we slow down these cases? So you've seen the uptick in cases, you've seen a lot of the response in the quarantine measures that have occurred. And so there's several ways that we can slow down these cases, which is really kind of what the country has been working on right now. So there's social distancing, which I'm giving us grades on the side of this, at least on my close family members and relatives. I think we're pretty moderately successful at social distancing. I think people are getting the idea that you have to stay away from other people. But as I go to work now, as I see people in the streets, there's not a lot of social distancing that's still occurring. I think we're getting a little tired of what's going on at the moment, which I understand, but it's really important to give good social distancing measures. The other things are washing hands, like we all know, are good infection control for the winter, keeping away from sick people, and working from home as much as possible. And I think we're getting better at these things. There's also a question about whether wearing a mask is important or not. We don't really know. I know that the recommendations are to wear a mask when you go outside and when you go into other businesses, especially food shopping, and everyone should wear these cloth masks. And the important thing about the mask is that they're not really protecting the regular cloth ones you can make at home or you buy. They're not really protecting you from getting infected from other people. what they're doing is limiting droplets from you spreading the virus to other people. And so even if, as I said before, even if you're asymptomatic and you don't have any symptoms, you still can shed virus. So these masks are really the, a safe and easy way to limit the amount of virus that you spread to your family members and other people around you when you go outside to businesses or cluster with other friends. And the other thing that we need to do is really test more. And I know you've heard a lot about this in the news about testing, and I just showed you the different kinds of tests that we're doing now. The number of and the efficacy of these tests is quite poor at the moment. There are, even though that we've tested 5 million, there's been 5 million or so tests in the United States over the last 4 months. We need that to double or triple or quadruple if we're going to have any real idea of where the cases are in the United States. And it's been a problem trying to basically follow this this virus as it moves, because we don't really know where it is, and the testing is just not up to where it needs to be at the moment. The quantity of tests, the speed of the tests, and really getting it out from just hospital settings to really the community where it can be used on a person-to-person basis. And all of these measures go into this idea of flattening the curve. And so I just want to go over really what this flattening the curve means. I know it's been used in the news a lot, but the general idea is that without any of the protective measures that we have accrued, whether it's social distancing, wearing the masks, quarantining people, staying at home, closing schools, without those measures, we would have clearly had, as was seen in Italy and Spain and China, this mass cluster of cases really going around the country, if we're just talking about the United States, in our communities. And we'd have a huge spike in cases where that overwhelmed the hospital capacity. And this would have caused way more deaths than we have, many more infections. It would have caused our doctors and nurses and healthcare members to have much more disease and then get infected and in an uncontrolled response. And this has been the big worry of why we need to lessen the number of cases in the country, in our community. And the idea of flattening the curve is that all these other social distancing measures and quarantine really lower the amount of number of cases and the burst of cases and the surge at the time, but really flatten this idea. They reduce the number of total cases at any one time. And I know it's hard to, there's no way to quantify without protective measures, what this number would be. And there's a lot of questions about, in the news now about whether these measures are really needed. And personally, I'd be much happier for people to complain about there being too few cases than too many cases. And there's really, there's only models that can predict how many number of cases there could have been. Clearly there would have been many, many more cases and a much surge of cases than what we know of right now. And so all the protective measures are doing well. I think that where we are in this current outbreak is kind of maybe at the peak of this case in the United States, somewhere in this range. We don't know how long this number of cases per day is going to last at this high level. But hopefully with more social distancing and continued quarantine, we will have a better idea and be able to reduce the caseload and number of cases in the community for over the next week to a couple months. Okay. So the next question is what therapeutics are being tested and which ones work. And so we'll go over this kind of through what we're doing. And so what's clear is that before this current outbreak of this coronavirus, there were no approved therapeutics that we knew of. So no FDA-approved drugs, vaccines, or antibodies were available to patients where they could, or doctors where they could treat anyone with it. It's for a variety of reasons. These were not approved and not developed, but they were in the lab. They're in my lab. They're in a lot of other labs, and they were never clinically gone through clinical trials for basically a funding and interest reason, to be honest. There are a lot of other therapeutics that are being tested right now, including, you've heard in the news, remdesivir, which is this Gilead drug, which has been used around the world now with kind of different results in different places, whether the patients are severe or not. You've heard a lot about chloroquine, which works in the lab quite well, but in people, not as evident that it's doing much in people. And there's a whole bunch of other drugs here, which we'll go through. But I think the important thing is that there's a whole series now of therapeutics that are being tested. So the first thing that we're doing in my lab and we're doing, the world has been doing, is looking at repurposing FDA-approved drugs. So things that are approved, we know how to dose them in people. They're approved for other conditions and other diseases, but maybe they can work against this new virus. And so that's what's being used in a lot of cases and a lot of clinical trials around the country, both here in Maryland and elsewhere. The other angle, the other level of therapeutics is to look at new antibodies that are being produced. So there's a lot of companies that are developing antibody products that are specific to this new virus. And so they can be used as therapeutics as well once they get through approval. So FDA-approved drugs can be used right away. Antibodies take a little while longer to validate and to prove they're working. The other kind of antibody product is something called convalescent sera, which is basically antibodies from people who have been infected with COVID-19, but then recovered. And so you can take the antibodies out of those people, and then you can give them to someone else to protect them. And this is in trials now, both here in Baltimore as well as around the world, to see if it really works. And there's evidence that it may be working. It's not really scalable to the number of people that need it. So there's kind of a cost-benefit ratio here that are important. The next thing that will take a little longer, but are vaccines. And so you've heard a lot about how vaccines will take 12 to 18 months before they really are widespread. We'll talk about vaccines in a slide or two, but that's true. It's going to take a while to get these vaccines through trials to make sure they're safe and effective. And we're working with a vaccine company here in Maryland. They're going to start their trial in humans in May in Australia, where there's a low number of cases at the moment. So they can do safety trials there before they get into people, into widespread populations. The other idea is that there is immunomodulators to really reduce kind of the severe disease once people get infected, as well as identifying novel drugs and inhibitors for the future. And so I'm just going to give you a couple slides of what we're doing in my lab. So what we've done previously was look at all FDA-approved drugs and to see how they blocked these two other coronaviruses, SARS and MERS, before this outbreak. This is just a paper we published before, and we identified about 24 FDA-approved drugs that inhibited both of these previous viruses, SARS and MERS. And so what we did when this virus emerged was we took that list of drugs and we tested them against the ones that worked against SARS and MERS, and immediately tested them against this new coronavirus. And what we found was that most of the drugs we found before that worked for the other coronaviruses now work for this coronavirus. And so you don't have to know what these curves are, but basically they show that a bunch of these drugs work against this new virus. And what we've done now, past the initial study, is we've looked at around 500 or so repurposed FDA-approved drugs to see if they inhibit this new virus. We're doing a lot of drug screens now through FDA screening for companies and other people on campus who have drugs that are maybe effective against their disease and looking to see if they work against this new virus. And what we really want to do is identify drugs that work against a lot of different viruses. So these are called these broadly acting antivirals. And so remdesivir, the one I talked about before, works against Ebola and Marburg and all other coronaviruses. And so what we're doing is really screening all these drugs now to look for their ability to work against a lot of different viruses. So we can have them basically in the clinic as rapidly as possible for not just SARS-2, but really for what happens in the future in the next outbreak. And then also in my lab, we're testing these in petri dishes against cells, and then putting into mouse models to see if they really work in animals as well. So we talked a little bit about vaccines, and I want to go into just a couple ideas of really what vaccines are, how they work, and where we're going with them. There's a lot of vaccines that are being tested in the clinic now. You've heard about one that the NIH is doing a trial for called from a company called Moderna. This is an RNA-based vaccine. The one we're working on is the company called Novavax here in Maryland. And there's a bunch of other ones using different kinds of basically ways of vaccinating people. And all of these need to be tested in people. They're not ready for massive rollout. They need to be tested, know they're safe, know they make really good antibodies against this virus, and whether they'll protect us in the future or not is what we have to figure out. And so just a general idea about how these vaccines work. Normally when viruses enter cells, this is the cells in your lung here on the left, they replicate. The little red dots so the circles here are the viruses replicating in cells. your body makes an immune response to these viruses, and it recruits immune cells to your lung to respond to this infection. And as part of this response, you get things that are called B cells and T cells responding, and they make these, then eventually they make antibodies. And these antibodies take about 2 weeks to make once you're infected with a virus. And the way that the antibodies work once after you've been vaccinated is that this is just a picture for a rhinovirus. Normally what you would get is you would get your virus infecting cells. Your antibodies would be bind to the virus specifically, and then they block the ability for the virus to infect your cells. And this is what we're trying to do with the vaccines that are being generated. All these vaccines are trying to make those antibodies that bind to protein on the outside of the cell, so that, outside of the virus, so that it can block the infection in your cells. And that's what all these vaccines do in general. But specifically the vaccines here want to target that lollipop-looking spike protein on the outside. Okay, so the next question is, when can we go back to normal living, which I know is quite important. And so this was a graph I pulled off the web, and I think that it kind of shows what I've just talked about before, is that in this red line here is what would happen in the number of cases over time if we didn't do anything. And so we'd have this huge spike in cases. In green is what we're doing now with all the preventative measures, where we're kind of at this beginning of this peak of cases and the daily occurrence of new cases. And so what we really want to do is to continue this decline in cases in this green line over time. And so over the next couple of weeks to a month, we want this line to keep going down on the new daily cases that are occurring. The big worry, though, is that as people go out, as people are kind of stuck at home, my parents are going crazy at home, my kids are going crazy at home, trying to manage this new situation that we're in. People start going out and kind of removing themselves and limiting their social distancing measures that were occurring right now. And if we do that too soon, we see this really big resurgence of cases. Even still, once we release all of these measures, we may see a resurgence of cases in the fall. And that's one of the big worries, is that there are, as I said before, asymptomatic people who can be positive for the virus, not know they're sick. They can go out to the the food store and go shopping and maybe spread this virus to their other community members, and we'll get another burst of cases. And that's really where what we're worried about now, and we're focusing on preparing the community for these future outbreaks, and especially what will come back in the fall. And so the state of Maryland has 4 basic steps that need to happen before all these measures are released, or at least slowly released. One is expanding testing capacity, which is going to be going right now. As I said, University of Maryland here in the Institute of Genome Sciences is charged with doing up to 20,000 cases a day. 20,000 tests a day. We need to still increase our surge capacity, which is happening as well around the state. The Baltimore Convention Center just opened on Monday with extra beds there in case we have a surge of cases. We need to ramp up our supply of PPE, which is personal protective equipment. So this is what all the hospitals wear, the masks and the gowns to protect all the hospital workers from getting infected. So in case there is a surge, they are protected and not overwhelmed. And there's something called contact tracing, which is where anybody who does get infected, they then, everybody that they've interacted with in the past 2 weeks also gets tested to see if they are now positive and don't realize it from that initial person. Contact tracing is hard, especially on a magnitude that this outbreak is occurring. And that's going to be much harder to deal with in our communities. And it's something in the future that we have to figure out how to do. And our kind of number 11 bonus is, when will it come back this winter? And so this is a graph of what the other 4 coronaviruses, the human ones that we get every winter, look like. This is data from 2009 to 13, but it looks the same every year, where we see these winter peaks of these cold viruses that you all know of every winter. And so we don't know if this new virus is going to have this seasonality like we know the other human coronaviruses do. Will it go down in the summer in large amounts? I don't know. It definitely doesn't seem like it's going away anytime soon. And one of the big questions is, if we do get this diminishing and we go past this peak of cases and we diminish to kind of a baseline level for the next several months, will we see a spike in the winter again when both kids go back to school, but also with the temperature gets colder, the humidity drops down where these viruses can transmit better. And it's unknown what the future of this virus looks like. I have no good answer. I think that my best guess is that it will diminish a bit over the summer, stay here through the fall, and then peak again in the winter, and we'll be back to potentially the quarantine social distancing measures that we're on right now. Okay, so what I hope I showed you was that this new virus, SARS-CoV-2 or COVID-19, is this newly emerged virus that we didn't know of before. That it is spreading rapidly through people. It's transmitting just like those cold viruses that I showed you on that previous slide. That increased age and comorbidities really impact the severity of the disease, which is a big problem for some of our communities in the country. That testing is really important. We're behind the 8 ball on testing at the moment. And it's a big problem for both the PCR positive and the antibody testing that is going to be rolled out soon. The therapeutics are getting through the testing phase now. There's a lot in the clinic in clinical trials. They really have to be vetted and validated to know whether they work or not, and that's what's ongoing. And then at the moment, we really have to continue these stay-at-home measures. I know it's hard to do all of these things. I have daily calls with my mom and dad talking about what they are doing or not doing to listen to my directions on a daily basis. They generally don't listen very well to me, so I understand that people are getting a little stir crazy at home. But I think it's too early to really let go of these measures, and we have to be really aware and careful about how we interact with our community for a little while longer. And so the last little bit here is I do have to thank my wife and kids. So my wife's a doctor at Hopkins. Her name's Jill Farner. She's a clinical geneticist. She has been doing telemedicine from home while my 10 and 7-year-old have been trying to do school. And so they made this big poster for my lab as we've been working pretty much 7 days a week since early January on this virus. And so I thank her and all of you for really taking care of all of us as we try to work on this virus, both my lab and in the hospitals. So I'm going to end there. I know there can be a lot of questions. If you have any questions, either you can type them in the box, you can unclick, you can raise your hand on the computer and you'll be unmuted. And I'm happy to answer any questions that I can. Thank you.
Thank you, Dr. Frieman. Again, I know it's been very busy and hectic for you, so thank you again for providing this much needed update. We really appreciate it. Again, like Dr. Frieman has said, if you're on your phone, you can press *9. From your computer or Zoom app, you can raise your hand by choosing the raise hand command.
Okay, so a couple questions coming in. So are there any ideas about what's causing these blood clots and stroke? And so that's a really interesting question. And I will say, I'm going to tell you what I think, but the real answer is we don't know. What we think is going on is that we know that this virus binds to a protein on the surface of cells called ACE2. So many of you may take ACE inhibitors for blood pressure control. And so the other thing that these ACE proteins do is they affect your coagulation pathway in this renin-angiotensin system. And so potentially the way that this virus is interacting with this protein on the surface of cells can affect the way that blood clots and some proteins in that pathway. And so maybe that's what's going on here, especially in the people who have really acute disease. And so a lot of little clots can form in the lungs during infection, and if they get released then from the lungs, they can travel and cause stroke. And so maybe that's what is going on. All of that is really kind of in the midst of what we're understanding right now. A question about whether a vaccine for COVID-19 can protect against the common cold. I don't think so. So the way that the vaccines are designed and the proteins on the surface of different coronaviruses are quite unique. They do share some things in common with each other, but generally they're pretty specific. And so unless the vaccine was designed specifically to really target other coronaviruses, it's probably only going to work against this new one. But I think that's yet to be determined, and there are people who are looking for pan-coronavirus vaccines that work against multiple coronaviruses. So the next question is by, is that Arnie or Amy? is really a good one. It's the question about if you've already been infected, can you get infected again? And we'll go to the second question in a second too. But so I presume, and what we believe is probably true, which is that if you are infected previously and you have antibodies to this virus, you probably can't be infected again. We don't know that to be for certain yet. It's probably true, but I think that we still, there's a lot of questions of whether that's true or not. And so there's some evidence in other countries and a small number of people where they got the infection, they went to the hospital, they got sick, they eventually got released, and then a couple weeks later, they came back with infection again. It's not really understood about what that means and whether it's an antibody issue for them or not. But I would bet the majority of the population, if you get infected once, you're going to be protected. But it's really yet to be determined what that really is, if that's true or not. And then the other question on that one was if the spread appears dormant in the summer, can older individuals return to going out? And so I know this is a big question, and it's really the question on all of our minds is if the virus kind of goes down during the summer, does that mean we can all go out and about our business again? And I hope that's going to be the answer. I think that that will be a slow roll. I think the important thing is that we have to still continue our social distancing measures. So if businesses start opening, if restaurants open, if food stores open more, if you go into those places, you still wear a mask, you still stay 6 feet away from everyone else, you still continue those measures, but you can maybe be slowly released kind of out into the wild again and allow yourself to go out in the community. And so all of that's really going to be determined by the number of tests and the number of really responsible individuals that are leading these businesses. Question about whether you were infected previously with SARS, the original one. Does it protect you against SARS-2? And there really is there's not, the evidence in China, which is where the majority of those original SARS cases were, is that you're not protected against this new virus. So the outside of the spike protein on the surface of the cells is quite different between the two viruses. And so it doesn't look like they can protect each other. Question about pools. And so whether, other than the social distancing measures, can swimming pools be opened again? And so I've got this from a lot of friends and family as well. If you can social distance yourself in a pool, I would think that's fine. There's no reason like water isn't going to transmit this virus between people, especially with chlorine in the water. But if your pool looks like anything like my neighborhood pool, if we say it's open, there's going to be 100 people there and it's going to be packed. And so I don't know how to balance social distancing measures in that circumstance with opening these pools. And I don't know what the right answer is. Maybe limiting the number of people in the pools in general. You still have to maintain the social distancing issue as we go.
Dr. Frieman, we have one question, one hand is raised from Miss Alana Basico. I'm going to mute you now. Please go ahead. Ms. Basico? Okay.
Okay, I'll just keep going.
Yeah.
Jump in if you have questions. So the next question from Tamika is about summer camp. And so just my kids go to summer camp as well in Baltimore and sleepaway camp. I don't know. I don't have a good answer about summer camp this summer. I've been talking to some of the people at these camps to help them kind of talk through the numbers about what's going on. I think that the ability to go to camp is really going to be determined on how fast the drop in cases are in the area where the camps are. One of the big worries would be that, can you quarantine kids at camp? My kids go locally to Beth and they go to Camp Area, Camp Louise, sleepaway camp, where I went as a kid. I don't know how, if there is a positive case at one of those camps, the camp will basically, my view is probably have to shut down. People will keep their kids home anyway. And I don't know what it means for camp this summer. I think it's a big worry. my kids are dying to go to camp, just like everyone else is. And I do worry that there's a bit of a problem with them being infected and then coming home and spreading the virus to their friends and family. A question from, I think this is Dr. Baskakov here at Maryland. What is known about the interaction with CD147 and brain infections? And so there has been some neurological issues with this virus. There was an early report that this protein called CD147 was involved with entry. I haven't seen any follow-ups or real papers other than an initial preprint of whether that was true or not. It doesn't look to be a neurological virus. There's probably other kind of immune response issues that are important for the neuro part of this, but again, it's yet to be determined as we figure this out in more people. And the last question there is, how soon can this person over 60 years old feel safe about returning to the workplace? I think this is my friend George on campus here. And I think that with the correct social distancing measures, I think people can return to work soon. I think that the number of cases is still quite large in this area. And the worry is that if you start really allowing people to come back too soon, there will be a huge burst in cases. And so kind of a measured, slow level release of people back to work is potentially warranted and soon, but I don't think we're there yet. One of the questions here is about the Pikesville area and these clusters of cases. And so and whether they're at senior facilities or not. And so I've been looking for that data actually. It's hard to figure out where exactly the cases are. That's just a HIPAA regulation about, you can't really figure out exactly what that means. There is a huge worry about about senior centers and senior facilities and senior living areas around the country. There's been a really large number of cases at various senior areas around the world, really, especially in the United States. And so that's a big concern. If you have a highly susceptible population that is able to be infected by this virus, they have little resistance to it, and it can spread very fast and very rapidly. And so especially at our senior facilities and senior centers around the country, that it's really the first level of concern of where this virus can spread and really cause rapid disease and death, unfortunately. One question about the antivirals. And so these broad spectrum antivirals, the one remdesivir, which is the one that is being used in people right now from Gilead, it targets the machinery that the virus uses to make copies of itself, kind of its copy machine protein, the polymerase is what it's called. That's what that drug targets. Various other broadly acting antivirals target different parts of the virus. The ones we're doing in my lab actually target our cells, so proteins that our body uses, that the virus uses from our body to replicate, and a lot of different viruses use those same proteins. And so we're targeting the host side rather than the virus side. And so both of them can be useful for developing antivirals.
Dr. Frieman, we have one hand raised. I'm going to unmute her, Miss Annette.
Yeah. Can you hear me?
Yes.
Okay. Yep.
I was just wondering, when you talk about the need for more prevalent testing, if you relax all of the restrictions on people and then you start testing, are you then, if there's a positive test, are you going to then enforce the restrictions again? I'm not sure what the testing is, how the testing is really going to help.
So first, I can't be blamed. I'm not the one that makes these rules, but I will tell you what the tests mean. So the idea is that as we ramp up tests, you want to do this idea called contact tracing. So if we release the social distancing measures and people are, go back to work in a small amount, certain levels of different shops open, people start shopping again. If you go get infected, if people go back out into the community to shop and to visit these places, if they then become positive, what we really want to know is who have they interacted with in the past 2 weeks, which is this kind of incubation period for this virus, or realm that they could spread the virus time period over the last, maybe 10 to 20 days outside of their own house. And the idea is that we need this mass testing so that we can do this contact tracing and really identify the people that they've been interacting with. And so kind of they both go hand in hand. It's knowing who you've been interacting with, but also being able to test them at really rapid and high scale. So you can catch the cases before they go back in the community. So like if we met each other at a store, and then 2 days from now, you have symptoms and you get tested and you're positive, one, if we don't know each other, then you'll never be able to find me. But if somebody puts out a report that says, you know, so-and-so was positive case at Eddie's on Roland Park on Tuesday the 12th, then if anyone had been in the vicinity of that area on the 12th, you know, contact your doctor for testing. You know, that's the kind of tracing that needs to happen. And it's a problem. It's a difficult thing to do. It's still obviously very disruptive, but we need this kind of tracing to go along. And so the Morton Katz has a question about what is the Sweden approach. And so the Sweden approach is basically started out as everyone being a good citizen, which is, and you know, kind of not making rules about social distancing, but making everyone aware about their ability to spread the virus. And so kind of being smart citizens in that case of not being around people so closely and not and trying to limit the spread. And so that worked for a little while. If you look at the numbers, I didn't put Sweden on that graph before, but if you look at the numbers, they started out kind of at a low level burn. And now what they're seeing is a really uptick in cases. And so there is this idea that you want to get everyone infected as fast as possible. That's what the England approach was early on before they looked at the numbers. And so if you do that and you say, okay, the best way to do this is develop herd immunity. Everybody should get infected. So you have antibodies against the virus, and then you diminish the amount of virus in the population. If we do that and you allow everyone to be infected just like that, what that means is that we know there's a huge population in the country that is highly susceptible to lethal disease from this virus, right? It's people that are elderly, people that are other immunocompromised diseases, diabetes, obesity, heart disease. You basically allow those people to be massively infected and have severe disease, and ultimately our number of fatalities goes through the roof. And so, you know, as not an MD, but a scientist, I don't really want the number of deaths to rise unnecessarily. And so there is a balance between the economic hardships and the health concerns and public health response. I understand that. I personally would rather the number of deaths not be as high while we continue the social distancing measures as we are right now. And so that's the Sweden approach, that you want everybody to be affected as fast as possible, so you get the mass number of immune people really rapidly. But that leads to a lot more severe illness at the same time, which is not, I think, you know, a smart approach.
Any additional questions?
Anybody else?
Okay. Oh, yes, we have one.
Here's one. What do you think will happen in the states that are removing social distancing rules? So in Georgia right now, they have, their case number is still going up. They actually have one of the largest number of cases in the United States outside of New York. And I think as they release the social distancing rules and people get tired of staying home, we're going to see large numbers of cases in these communities. I just think that's exactly what's going to happen. And so the longer that we can go to kind of slowly release people from these quarantine measures, but in places where they are seeing a low number of cases in the community first. I mean, you have to see the downturn before you start letting people out. If you let people out when you're still rising, the numbers just are going to go through the roof. And that's really the worry. And I think that in the areas where we are going to see, where they're releasing these people from social distancing and quarantine, we're going to see a large number of cases. I think you'll see it. You're not going to see it tomorrow because those in Georgia and those other states, they just started this week. So in 2 weeks from now, or 10 to 14 days, you're going to see a really steep rise, which are the cases that are getting infected right now. Let's see, there's a couple more coming in. So if all cases disappear and one infected person comes in location, will this start all over again? So if you have one person in the location, it doesn't mean that everybody gets infected, but obviously the probability of that person infecting more people is higher than there being no cases there at all. And so everybody spreads virus differently. There are some people who spread it at very high levels and some people who don't. Every person has kind of a different spreading phenotype. but there's that, the more people that are in an area with that are positive and spreading virus, the more number of cases we're going to get. And then the last question on there is, if you test negative, are you still susceptible to the virus? And so if we had a really good antibody test, which we don't at the moment, but if we had a really good widespread antibody test and you were negative, then you still are probably susceptible. You're susceptible to the virus, as you were 6 months ago. If you test positive, what we don't know yet is if you are now resistant to the virus, and that's just something that we don't know. And it's going to be basically a worldwide experiment to know how much antibodies you need in your body to be protected from infection. And we don't know the answer to that, and it's not something I can identify in the lab. It's something that we need to figure out kind of in the population level.
Thank you so much, Dr. Frieman. We appreciate it.
Thank you very much. Good luck, and stay safe.